Thank you very much, Alex. So I'll try to make this as short as possible because we know that we are late of time, but it's really very important to understand, the path for biosimilars and how biosimilars, I think, and we at ESMO think that it's really very important to introduce them in the clinical setting. So these are my conflicts of interest. So basically, the bottom line here of this slide is the fact that in the next years, actually, also in the past years, several biologics have expiration of the patency. And you can see that the lines are different from the United States and Europe, but and also in different countries. But the important message here is now we have the time where some drugs that target malignant cells or target malignant processes from our patients are under the process of expiration of the patency. So now we have the opportunity to discuss how biosimilars may help in several aspects to make our health system sustainable, but more importantly, to provide opportunities for patients to have access to drugs that in some countries they are not accessible. So just a couple of slides on the financial burden of the tumor burden. I think that all of you are aware that in the last years, and these are global numbers, the cost of treating patients with cancer is increasing. And you can see here, for example, that if we compare the data in 2010, and this is, in 2010, and now we see the expected data in 2020, you can see that there is a clear increase in the cost. And it's estimated by 2020, the global cost of the drugs are going to be around 150 billion. Another important figure is that at the average in Europe, the cost of cancer drugs compared to the cost of total cancer care, and total cost of cancer care means prevention, diagnosis, GP, and obviously hospitalization, emergency, and also the cost of the final life. So the cost of the drugs at average is 30% of the total cost, so 30%. So this is an important figure. But also we know by different surveys that we have done at ESMO that accessibility to drugs that are approved, for example, in the European Union, it's very different depending on the income of the countries. And in this slide, you can see that the access of some drugs for lung cancer in Western countries in Europe was quite accessible in most of them, but actually if you look at the data in the eastern countries, you can see that there are many countries where you have red spots, black spots, meaning that these drugs are not available, even they are approved, because they are not reimbursed. So basically, as mentioned before, I think that there are two important aspects. One thing is to make our health systems sustainable over time, and therefore be able to implement and to have more innovation with the new drugs that are really active. But at the same time, we see that there are many countries where access to these drugs is very difficult. So obviously, most of the regulatory agencies have been working a lot on the definition of biosimilars. EMA was the first one, but if the FDA as well, and also some agencies from other countries. So this is really very important. I'm going to go perhaps to the more practical definition of biosimilars that we introduced in a position paper that the Executive Board of the European Society for Medical Oncology did in 2016. And actually, as you can see here, biosimilars are medical products containing a similar version of the active substance of their originator or reference product, that usually it's a biological drug, derived from living organisms. And this is really very important, the fact that they are derived from living organisms. Importantly, when we talk about biosimilars, people always think about monoclonal antibodies because this is the area where we have more opportunities. But actually, it's not only about monoclonal antibodies. This relates also to hormones, vaccines, small proteins, fusion proteins, and other biological products. So I really think that this is a good manuscript, and if you have the time to read that, actually offers very important information on several aspects of the development and the uptake of biosimilars. Importantly, also, to realize that sometimes there is some confusion about the wording And it's important to say that biosimilars are very well defined, but you have to separate that from, for example, for biobetters, that it's a different thing. It's try to improve the versions of the original biologics, but more importantly, to separate that from what we call biomimics or biocopies, because these are compounds that have not developed with biosimilar regulatory pathway, and this is something that it's important, especially in some Asiatic countries. So obviously these compounds have not the same standard procedures and the standard evaluation that biosimilars have. And obviously generic drugs relate to the field of small molecules. And actually this is important to understand the magnitude of the problem sometimes in comparing small molecules and biologics. So you can see small molecules are usually very small as they are defined, and they are at the time that we talk about the compounds, they are generics and they have exactly the same structure. But in the case of biosimilars, this is not the case. There are small differences between the originators and the biosimilars. But actually, these differences are also present between different batches of the original compounds. And I will come later on to this because this is really important. So when you look at the monoclonal antibody, for example, the FAV portion is identical from the original and the biosimilar, as well as the FC portion. Where we see some minor differences are in the number of polysaccharides that the monoclonal antibodies have in this central part of the antibody. And this makes a little bit the difference between them. Also, you have in here the separation between generic and chemical drugs and biologics and biosimilars. Perhaps we have touched about all these points except for the last one, the point of immunogenicity. And this is really important because, as mentioned, the small difference that we can have between the different batches of the original drug, but also between the biosimilars, can produce immunogenicity. So what is immunogenicity? How we can define that? Actually, this is the ability of a particular substance, such as an antigen or an epitope, to provoke an immune response in the body of a human being. And the ability, of course, to induce a humoral or cell-mediated immune response. And actually, this relates a little bit to the process of manufacturing that I mentioned before. But again, this is also present between different batches of the original drug. It's also important to mention that the development of biosimilars is much more complex than the development of generics. So you can see here in different areas and aspects how important is the time, the cost, and the manufacturing process, more similar to the biologics than to the generics. And perhaps I can skip this slide. But the important thing here is that the regulatory authorities do have control of all the process, and actually they look at the follow-up and the monitor of these drugs in a tightly regulated process, just to ensure that there is consistency between the different batches. And again, this consistency is the same that it is asked and looked for between different batches of the original drug. Obviously, being said that, the clinical development of biosimilars compared to the originators are completely different. So if we start with the originators in the left part of the slide, you can see that most of the resources and most of the investment is being projected at the time of clinical development, the clinical studies. But actually for the biosimilars, it's the opposite. You can see that this is the pyramid and the inverted pyramid. For the biosimilar development, most of the studies are done preclinically, both in vitro, in vivo, and clinical pharmacology as well. And then we have less clinical studies. Because actually, when we talk about the regulatory label and the approval of biosimilars, usually the regulatory authorities and the pharmaceutical companies decide an indication, and this is the indication base for the biosimilar, and they, based on the indication, they do extrapolation to all the other indications. So you usually have to demonstrate the activity of a biosimilar in one single indication with a sensitive endpoint. It may be overall response rate. Usually this is the one that it's used, but it could be also progression-free survival or overall survival. But usually it's used overall response rate. And then once you have data supporting the similarity in one particular indication, you can extrapolate that biosimilar to different indications based on the scientific rationale, but also on the way that the biosimilars are developed. And actually, if you look at the pathway on how the different regulatory authorities have contributed to create models and to create the procedures for developing biosimilars, you can see that this is a long time away, started by the European Union and also EMA back in 2006 for the guideline of clinical development of biosimilars. But you can see that more and more countries are actually having their own biosimilar regulatory pathways introduced. So important things about the biosimilars, the labeling. What should the labeling include in a biosimilar? First of all the information about the sensitive population that has been used to demonstrate the biosimilarity. And this is important just to look at the endpoints that are used. Number one. Number two, the pharmacovigilance plan. As mentioned before, it's very important that we look at the follow-up of these compounds. And number three, something that is very unique from the biosimilars is that the brand name of the reference product should be mentioned in the label. And also, it's very important to have comprehensive data about all the extrapolation process, but also some of the aspects that we're going to be defining in the next slides, like interchangeability, switching, automatic substitution, and immunogenicity. So really, this is very important. And also, it's important to involve all the healthcare professionals and the patients from the early beginning. This is really very important. A couple of concepts that we can summarize. Substitution is a concept where the pharmacist may dispense an alternative biological drug for the prescribed biological drug. Automatic substitution, actually it's more a force to do that by the regulatory authorities or by the national health systems or the payers. Switching is and interchangeability are a couple of concepts that sometimes are used in the same way, especially in Europe. And basically, this means when a prescriber change also with the consent of the patient from one therapy to the other. And actually, these concepts are not equally introduced by the different regulatory authorities. For example, Canada, Japan, globally, the United States do not support automatic substitution. Actually, the European Union do not make any statement on that. They refer to the competence of the national authorities to decide on that. And even some other regulatory authorities have not positioned themselves on that particular area. Importantly, as I mentioned before, the position of ESMO is that extrapolation is acceptable, provided that we have the rigorous data on the first indication with the sensitive population and the sensitive endpoint. But importantly, all analytical, preclinical, pharmacokinetic, and pharmacodynamic and clinical data, and also all the data on immunogenicity has to be reported and prospectively follow up. So in this case, with this particular indication and requirements in our means, extrapolation it's acceptable. What about switching? Actually, switching, as mentioned before, is the process of changing one compound from the other. But this is a decision of the physician and the patient, and something that has to be discussed between the two of them. And actually, it's not in one unique direction. So, for example, the obvious one is that the reference product at some time point may be switched to a biosimilar. But it also can happen that the biosimilar can be switched again back to the reference product or from one biosimilar to another. Our position, at least the ESMO position, is that automatic substitution should be avoided because it may create problems between the community and the patients. But the most important slide actually is this one. It's important to build the confidence between all the stakeholders, with the patients in the middle. So I have presented you before the opportunity of biosimilars to promote sustainability of the health systems, number one, and also to promote access to more innovative drugs in some countries for patients. But it's very important to have the complete view of all the stakeholders together, with the science behind, with the political guidance, with all the data collection and the data analysis and prospectively registries that are going to be providing this information. But as you have seen here, it's not only about the professionals. So professional societies are important, and statements from professional societies are really good, but the clinicians, the pharmacists, the nurses, also the regulators, the payers, the regulatory authorities, of course, but the patients in the middle, it's important that they discuss this from the early beginning. We can skip this slide. But actually, at ESMO, what we tried to do, just to understand which was the knowledge and potential actions that we could do in the level of biosimilars, we did a survey just to address several questions again on the knowledge and whether the patients and the physicians felt comfortable with the use of biosimilars. This is a survey that we presented in Munich last year at the ESMO Congress, but basically we had almost 500 respondents to the surveys, and you can see that it was really a global survey. Most of the respondents were from Europe, but you can see that we had physicians from all the places in the world. The important, in one slide, the summary is that we could say that 79% of the responding population actually had moderate to high level of the knowledge of biosimilars, number one. The level was a little bit higher, only a little bit higher among the European colleagues compared to the Asiatic colleagues. 49%, almost 50%, use biosimilars in the routine practice, and this percentage is really higher in Asia than in Europe. And around 50% of the respondents felt really moderately or very comfortable by using biosimilars, because in this way they think that we are promoting as much as possible sustainability of the health systems. So the last slide, you have seen that several regulatory authorities, including EMA, that was the pioneer regulatory authority for the use of biosimilars, have done a rigorous regulatory process for that. It's important to know the concepts that I mentioned before about comparability, extrapolation, and switching. It's also very important to collect post-approval data, and actually there are several projects trying to get as much as possible data for that. Collaboration, it's fundamental, it's critical with all the stakeholders. And again, if we implement the use of biosimilars in clinical practice in a proper way, actually we will be promoting the sustainability of the health systems and also the access to drugs to cancer patients in many places. Thank you very much.